Okay, so in this video, we'll be covering the last type of epistasis known as duplicate genes. Okay, so just to clarify, so far we have covered 4 types of epistasis: recessive epistasis, dominant epistasis, complementary gene action, and duplicate genes. There are more than these 4. Okay, so what we are introducing in the notes are 4 common ones, but there are others as well. Okay, so these are not the only 4 lah. That's number 1 and number 2, right? As the learning outcome has stated, there is no need for you to memorize the different types of epistasis and memorize their ratios in the phenotypes. Okay, so the more important skill is to look at the information given in the question and to deduce how the epistasis or how the masking is how it plays out, right? How is the mechanism like? And when you know how the epistasis is carried out or how it happens, right, then you would know what should be the phenotypic ratio due to the masking and the epistasis. Okay, so it's more problem solving based on information given that is required instead of memorizing the different types of epistasis and ratios. Okay, so we are only showing you like 4 slightly more common ones that we have seen so far in past year questions, but they can really give you like other examples that we have not seen before, but you just solve the question right, based on the information given. So they surely will have to describe some kind of interaction and some kind of masking in the information given in the question, right? So you just follow what is given. All right, so just to clarify that. Okay, now we'll go and look at the last example of epistasis, duplicate genes. Okay, so for duplicate genes, what happens is we have multiple enzymes or proteins, okay, that are involved in the same reaction and that give you the same phenotype. Okay, so in this in the example that we have here, we have 2 different enzymes, enzyme A and B, that both catalyze the conversion of the white pigment into the yellow pigment. Right, so in the presence of either active enzyme or both, right, all of that, right, either one enzyme or both will give you the same phenotype, yellow. Absence of both, or both enzymes being inactive, then that gives you the white phenotype. Okay, so in this case, right, when you cross the double heterozygote, right, the phenotypic ratio would be 15:1. Right, so looking at the diagram, instead of the 9:3:1, we have the 9:3:3 added together to 15 and 1. 1 is when both enzymes A and B are inactive, right? Then for the 15, either both enzymes active or either one, right? So enzyme A active here or enzyme B active here, right? All of them give you the same phenotype. That's why it's 15:1. Right, so this is duplicate genes, multiple genes, coding for different proteins that gives you the same phenotype. Right, so presence of either one or both, same phenotype. Absence of both, another phenotype. Okay, so that is duplicate genes. Very simple.